So let's now consider DNA replication. And as you know, this beautiful molecule is replicated in a semi-conservative manner, and we've already know that it is replicated in a semi-discontinuous fashion as well. And we'll be reviewing some of those concepts as well as talking about some of the macromolecular machines involved in DNA replication and the origins of DNA replication on chromosomes. So that's what we'll be talking about for the next little while. And let's start out by first identifying the major macromolecular machines present at a DNA replication fork. So let's look at some of the players, if you will, of the DNA replication process in cartoon form. We have DNA helicases, single-stranded DNA binding proteins, topoisomerases, primase, RNA primase, DNA polymerases themselves, associated DNA sliding clamps, and RNase H to remove RNA primers, and DNA ligase to seal nicks. So first, let's look at the role of DNA helicases. They use the energy provided by ATP hydrolysis to unwind the DNA double helix, and in so doing, provide single strands that can serve as templates upon which new strands can be synthesized. And this all occurs at the replication fork, which is the junction between separated strands and double helical DNA. Now, in order to prevent reannealing of the separated strands, there are single-stranded DNA binding proteins, or SSBs, that anneal to the separated strands in a fashion so as to prevent reannealing through Watson-Crick hydrogen bonding. Now, as unwinding occurs, positive supercoils are induced in the DNA, and since the linking number, that is the number of times these strands cross each other, remains constant, but the twist is decreased as DNA unwinds, the writhing has to increase as DNA unwinds, and we end up with supercoiled DNA, or positively supercoiled DNA, which needs to be relieved by the action of topoisomerases. And these topoisomerases use the energy provided by ATP hydrolysis to relieve the positive supercoils. And we'll talk a little bit about topoisomerases later in this lecture. Primases synthesize RNA starters, or RNA primers, upon which DNA polymerase can extend those primers. So the primers are essential for the action of the annealing of DNA polymerase and the extension of those primers by DNA synthesis. So every new strand of DNA starts out with an RNA primer and is elongated by the action of DNA polymerases. Now, as we've mentioned and discussed earlier, the replication of DNA is semi-discontinuous because of the exclusive 5' to 3' synthetic activity of DNA polymerases. And that leads to the production then of a leading strand in which DNA synthesis is continuous, often initial primer, and a lagging strand at which new primers need to be synthesized as more DNA is unwound, and DNA polymerase extending those primers leads to the production of Okazaki fragments. And it takes only approximately 1 second for DNA polymerase to bind DNA, but DNA can add new nucleotides at up to 1,000 per second. So DNA polymerase remains associated with its template as it synthesizes new DNA. And that association with substrate for each round of synthesis, instead of falling off the template and remaining associated with the template, is called processivity. And that processivity is endowed to DNA polymerases by processivity proteins. And these are known as DNA sliding clamps, or sliding DNA clamps, shown in purple here. And these subunits of DNA polymerase anchor the polymerase on the template and allow for processivity to occur. So here we see semi-discontinuous DNA synthesis on the leading and lagging strands. And this leaves a problem for the replication machinery in that the RNA primers need to be removed by an enzyme, and the enzyme used in E. coli is RNase H, which removes the RNA primers from the Okazaki fragments and from the initial template on the leading strand as well, the initial primer on the leading strand. And that is the action of RNA H. Now one nucleotide is left on, one RNA nucleotide is left on the DNA, and that needs to be removed by an exonuclease that is species specific. We'll talk about that a little bit later. And then what remains on the lagging strand are these Okazaki fragments that need to be extended after removal of the RNA primers by DNA polymerase. And in E. coli, this is DNA polymerase 1 that conducts this synthesis. And finally, DNA ligase, using the energy provided by ATP hydrolysis, seals the nicks in the sugar phosphate backbones, and we end up with a continuous DNA strand. Now, because we have a leading and lagging strand, we need two DNA polymerases at the replication fork. And we refer to those, to the doublet of DNA polymerases, as a holoenzyme of DNA polymerase. This is a multi-subunit protein, multi-subunit complex, which consists of DNA polymerases, the sliding DNA clamps, and a DNA clamp loader, sliding clamp loader that loads the sliding clamps onto DNA polymerase and allows the DNA polymerase on the lagging strand to synthesize a stretch of DNA and then fall off the template because the sliding clamp is recycled, and then synthesize a new stretch of DNA as a new sliding clamp is loaded upon it. And this is occurring continuously on the lagging strand of DNA synthesis. And this model is called the trombone model because the DNA is being pushed through the polymerase, if you will. The leading strand is being synthesized continuously, but the lagging strand needs to be looped out continuously for synthesis of each new Okazaki fragment from a new RNA primer. And that requires recycling of the DNA sliding clamps, the processivity factors, for the lagging strand polymerase. That requires loading onto that clamp and then disassociation from that clamp in a continuous process. And that is accomplished through a protein called the sliding clamp loader, shown as this horseshoe-shaped structure here. There are long extensions coming from the sliding clamp loader are another protein that associates the clamp loader with the polymerases and the helicase, and that is called the tau protein, as we'll see shortly. So this trombone model provides a means by which rapid synthesis of a DNA daughter strands from a parental DNA double helix can occur, even though we have semi-discontinuous synthesis occurring, that is, discontinuous synthesis on the lagging strand and continuous synthesis on the leading strand. So you should be able to kind of visualize this as a dynamic process in which the DNA is continually pushed through the DNA polymerases and their associated DNA sliding clamps, those clamps providing for processivity. But on the lagging strand, it's like a trombone being pushed through, the slide on a trombone being pushed through the DNA polymerase. Note the action of RNA H removing the primers continuously on the lagging strand. As each new primer is synthesized, the RNase H removes that primer, and DNA polymerase extends the DNA. So now let's look, or let's refresh our memory and remember the actual catalytic event, the biochemistry that's being done by DNA polymerases, the catalysis that's being done by DNA polymerases as they synthesize DNA. And then we'll move on to a static view of some of the molecules we've just looked at in cartoon form. And then finally we can get into some of the wonderful details of the macromolecular machinery that is conducting this process. So let's look at the incoming nucleotide for DNA synthesis. As you know, deoxynucleotide triphosphates are the substrates which are acted upon by DNA polymerases. And we have a nitrogenous base, in the case of DNA, A, G, C, or T, a deoxyribose sugar, which does not have a hydroxyl group on the 2' carbon, but does have a hydroxyl group on the 3' carbon. That's why we call it deoxy, because it's really 2' deoxy. And here is our first carbon, second carbon, third carbon, fourth carbon, and fifth carbon of our sugar. And then we have our third part of the nucleotide triphosphate, that is the triphosphate part. And we have an alpha phosphate, a beta phosphate group, and a gamma phosphate group. And it is, as you know, it is the alpha phosphate which is attached to the 3' oxygen on the last added nucleotide of a growing primer strand. So DNA polymerases then add nucleotides to the free 3' end of a growing strand, and they do so by catalyzing the nucleophilic attack of this 3' oxygen on the alpha phosphorus atom, and in that way add a new nucleotide with the release of pyrophosphate, which then gets degraded into inorganic phosphates. So here we have an incoming nucleotide. We have base pairing, ensuring that a correct nucleotide is inserted on the newly synthesized daughter strand, and we have AT-GC hydrogen bonding rules, of course, observed. And we have this nucleophilic attack on the alpha phosphate by the 3' oxygen of the last added nucleotide in the growing chain, the release of pyrophosphate that then is degraded, yielding a negative delta G, highly negative delta G, when that is degraded into 2 individual phosphates. And then we have now our growing strand has been elongated by 1 nucleotide. And there is a new free 3' hydroxyl group whose oxygen can attack the alpha phosphate of a new incoming nucleotide. In this case, it would be a deoxy nucleotide that would bear the base of guanine to match with this template strand C. So let's continue now by looking at revisiting the replication fork that we looked at before, and remember some of our key players here. So you'll remember the core of the macromolecular machinery, we'll call it the replisome, which is a macromolecular assemblage at the replication fork. The core of that consists of 2 linked, in the case of E. coli, polymerase 3, DNA polymerase 3 polymerases. And these are linked in complex with a clamp loader protein called the gamma complex, which loads sliding clamps on free polymerase so as to ensure the processivity of the polymerase as it synthesizes DNA. And this tau protein is a flexible linker, which allows some flexibility at the replication fork, because in the trombone model, as you know, there's a lot of movement going on. So if we look at this in a little more detail, then we have this the tau protein linking the clamp loader with the helicase at the core of the replication fork, and then the 2 DNA polymerases, one synthesizing continuously on the leading strand, one synthesizing discontinuously on the lagging strand. And if we look at this in static fashion, I'm not going to go through all these diagrams because they're there simply to re-emphasize the model that I presented before, the trombone model of semi-discontinuous DNA synthesis. But you should be able to identify all these molecular players and understand their role in the process of DNA replication, including the loading of the sliding clamp onto, repeatedly loading the sliding clamp onto DNA polymerase on the lagging strand. It must be continually loaded on there as we synthesize a short stretch of DNA and then reinitiate synthesis at a new RNA primer as the trombone slides through the DNA polymerase. So you can freeze the slide at this point, or freeze the movie rather, at this point, and study this some more at some point, and you're free to do that. So before moving on to the next part of this lecture, I just want to mention that we're going to have to consider some of the discriminatory mechanisms that DNA polymerases are endowed with, so as to ensure mistakes are kept to a minimum as new DNA is synthesized. And that's where we'll pick up with in the next part of this lecture.